Hi, Bio30, 16.2 today. Mitosis is the topic, and what this is cell division of body cells. And we know that all body cells have 46 chromosomes. So in mitosis, what's happening is that body cell is going to double its DNA and split into two cells, each with also 46 chromosomes. So that's what our topic is today, how our parent cell splits into two daughter cells that are exactly the same genetically as the parent. We mentioned here that it's important to do this when there's growth. So whenever cells or an organism is growing, it has to do mitosis for that growth to happen, for worn out cells to replace themselves or to repair like a cut on your skin. This all will involve mitosis. So for this to happen successfully, we have to take our DNA, and we'll look at it as a single strand here, and we have to make an accurate copy of it so that we have two copies of that genetic material, and then we have to divide that into our two cells properly for us to get two viable identical cells resulting. So what we're looking at is the four phases of mitosis called the PMAT stages, and those four are prophase, metaphase, anaphase, and telophase. And that's what we're going to focus on in this lesson, is what the main important events are in each of those stages. So what we have here is that in the cell cycle, that interphase has already happened, where the cell has doubled its DNA. The chromosomes are all now in the shape of an X, and we're just looking at that 10% of the time when cell division mitosis stages are happening. So the first significant stage, prophase, is when you actually see the chromosomes. It's hard to see in this picture, but they are in a shape of an X, so those chromosomes are all replicated, I should say. The next stage we're going to focus on is the metaphase stage, where they all line up, meet down the middle of the cell. So all, in human terms, 46 chromosomes would meet down the center. Sometimes we call that the equatorial plate or metaphase plate. Those chromosomes will meet down the center. The third phase, anaphase, will be where the chromosome is pulled apart right at that joining spot, the centromere. It's joined apart and goes in the two separate directions. And in telophase, you see the splitting of the two cells in half. Now, we're going to go through each of those stages a little more carefully and take a look at what they look like on just regular light microscope diagrams. That interphase that happens 90% of the time, basically what you see there is a nucleus with just a dark mass of chromatin in there. So 90% of the time a cell is just existing and doing its job as interphase and preparing its genetic material for a division by doubling it, which is called replication. The PMAT stages, first one is prophase, is when you actually see that it's significantly darker in the nucleus because you see those individual chromosomes appearing. So compared to interphase, where your chromatin is just like as a jumble, in prophase, remember it goes PMAT, P-M-A-T, in this prophase stage, you see the chromosomes clearly and that they are a shape of an X and they have replicated. So seeing them is what's significant in prophase, but also you'll notice that our nuclear membrane is disappearing here. So no longer will the chromosomes be in a nucleus. The activity of separating those chromosomes will take up the whole like kind of space of the cell, so you don't see a nucleus anymore for these 4 stages. So next one, so we basically see the chromosomes in prophase, and in metaphase we see the chromosomes line up down the center, down the metaphase plate. Basically they meet down the center. In an electron microscope or light microscope picture, seeing these chromosomes lining up down the center, you see this really dark band. So it's pretty cool seeing this under the microscope. It would look like that. The chromosomes line up down the center. And moving on to anaphase, we see that these little spindle fibers coming from the centrioles here. We have centrioles at the 2 poles. And those centrioles make these spindle fibers that actually grab on to the centromeres and pull apart, because that's where their 2 sides are joined of a chromosome, and they'll actually pull apart those chromosomes. And that is the significant thing about anaphase, pulling apart the chromosome. And the last one, telophase, is where the cell starts pinching in to form 2 cells. So we see cytokinesis, which is cell cytoplasm splitting happening. So you can see that it's splitting into 2 cells here. Telo often means the end phase. And you also see here the nucleus forming again, and it'll encompass those chromosomes. But what you started off with, whatever chromosome number, if this was human cell, you start off with 46 chromosomes in the nucleus. Those 2 cells that result at the end would be 2 of them that have 2 cells that each have 46 chromosomes in them. That cell that started off, like in prophase, the prophase cell would have 46 chromosomes that are in the shape of an X, whereas when we finish, we're going to have 46 chromosomes that are just 1 strand. Okay, so it still has all the material, genetic material, but they have split into 2 cells. So the notes in this just go through those events again more carefully. Interphase, we see that there's chromatin in there. In interphase, this replication is happening where chromosomes are replicating and making the other side, identical side. We see our prophase stage next, and in prophase, we're going to see that there's X-shaped chromosomes, and the nuclear membrane is coming apart. Those centrioles are kind of headed towards the 2 poles because they will be making spindles very shortly. So you can see the dark X's, chromosome and X shape in this microscope picture. Second stage is metaphase, and in this fluorescence method of microscopy, we see the chromosomes as a dark band here in the blue, and we see the spindles as this green section. Here's a cell. And so metaphase is when those chromosomes line up down the center. Our centrioles are on the 2 opposite sides. Those spindles are starting to form, and it will be in anaphase when those spindles start attaching to the centromeres of the chromosomes and tugging. So the spindles start shortening and tugging apart, pulling apart the chromosomes. So the chromosome would look like this, and in metaphase, but in anaphase, those little centrioles are shooting out those spindles, grabbing onto where this chromosome is joined, and it's going to pull apart the chromosome, and it would in anaphase end up looking like this. So it's pulling it apart, and then the 2 cells will each have half of that chromosome, and they are identically, have all the genes on them because it made a double of itself. And so the entire recipe will be in both of the resulting daughter cells. There we have the chromosomes getting pulled apart. And the last stage, telophase, is when our cell actually splits. Cytokinesis is when it splits into two and starts to form the nucleus again around the chromosomes. And so in telophase, you actually see that there's two cells forming. If they're animal cells, the roundish cells, the splitting in two is called a cleavage or cleavage furrow that's forming, kind of like a cleaver cutting a ball of dough. However, if it is an animal cell, what's going to happen, or plant cell, because of this thick cell wall that plant cells have, what has to happen in plant cells is that it has to make a cell plate, which is extra cell wall made of cellulose, between the two cells that are forming. So in an animal cell, you're just pinching it in two, and in a plant cell, you're forming this extra cell wall there. Cytokinesis, we talked about the cytoplasm splitting, mitosis in animals, mitosis in plants, and I think that pretty much wraps it up. It does say that if the cell cycle has some error, we can have uncontrolled mitosis happen over and over, and the cell kind of skips the interphase and doesn't become a functioning cell. So that's what cancer is. They're not differentiating and becoming functional. So last thing here, it talks about how studying cell division is very helpful in terms of studying cancer, or looking at how the cells can be studied and how cells grow. And there's a lot of technologies that depend on studying mitosis. In our lab, we would have seen the this whitefish slides on under the microscope, and this is basically images of what you would have seen. Interphase, you don't see anything, you don't see the chromosomes being dark, but in prophase you do see the chromosomes because they chunk up into those X shapes. In metaphase, you'll see the dark band down the center because they're lining up down the center. In anaphase, they're pulling apart by spindles, so you don't see the spindles here, but spindles somewhere are grabbing the two sides of the chromosome and pulling them apart. And then in telophase, you see the two cells pinching into two. So that would be in an animal cell, whereas in a plant cell, here we see the thicker cell walls. This is an onion root tip, which is dividing very rapidly. And you can see a mix of all these stages at one time. And that's really, in real life, what happens is you might see prophase, places where you see a dark chunky pile of chromosomes, metaphase, where you see the chromosomes lined up down the center, anaphase, where you see them in two sides getting pulled apart, and telophase, where you see a new cell wall starting to form down the center, and it'll form all the way across. So that's the two new cells forming. And remember, whatever the original cell had for chromosomes, if it was like a 40 chromosome cell, for example, each new cell that forms is also going to have 40 chromosomes. But as we begin mitosis, those chromosomes are the shape of an X, and the two daughter cells that form will each have half of it. And that's still the full set of information. There's a lot of really great little links here that you can check out on mitosis and see it happening with animations and real life footage. So check those out, guys. And thanks for listening to the mitosis stages, and we will catch you next time for meiosis.